Hello everyone, I'm Thiébault from the Vision Institute in Paris, and I would like to thank the consortium for giving me the opportunity to present the work of my team, which is impact of lactate transport deficit in age-related macular degeneration. As you know, the consortium performed a few years ago a meta-analysis of 14 genome-wide association studies that has included over 17,000 cases and over 60,000 controls. And at that time, they have identified 7 new loci, reaching the genome-wide significance of 5 times 10 to 8 for the first time. And it has included SLC16AA locus on chromosome 22. Since then, we have decided to focus on SLC16AA gene for the following reasons. First, SLC16AA gene was identified 20 years ago by Nancy Philip and her team. Also, the same team not only demonstrated that SLC16AA gene was encoded for the monocarboxylat transporter 3, which is a lactate transporter, but also that MCT3 transporter had a restricted expression to the retinal pigmented epithelium, as you see here, with the result of RT-PCR from different tissues from wild-type mice, but also a basolateral localization on the RPE, as you see here on the images of wild-type mice, but whose expression is suppressed in knockout mice. Then, we confirm the result by a bioinformatic analysis using gene expression omnibus. And also, we have shown that SLC16A8 mRNA has a restricted expression to the retinal pigmented epithelium. So, what is our working hypothesis? As it was previously demonstrated, the long-life renewal of corneal outer segments requires support from the rods via the rod-derived coagulability factor protein. This protein stimulates aerobic glycolysis to produce triglycerides or phospholipids, which are essential to build the corneal outer segments. But aerobic glycolysis produces also lactate, which is secreted by the cones and then transported through the retinal pigmented epithelium to the choroidal circulation. And since MCT3 is only expressed on the basal side of the RPE, we can say that MCT3 controls the transport of lactate out of the retina. And so, the discovery of AMD risk allele in SLC16A gene would imply that a deficit in lactate clearance on the basal side of the RPE will trigger here a deficit of lactate transport on the apical side of the RPE, resulting in accumulation of lactate in the interphotoreceptor matrix. This will reverse the polarity of lactate transport in the cones and finally counteracting ADCVS-stimulated aerobic glycolysis. So we think that this mechanism is likely responsible for the impairment of photoreceptor function and cone damages in SLC60AA knockout mice, where we have observed an increased extracellular lactate in the retina and an altered visual function. So we think also that this mechanism might be responsible for the loss of corneal outer segments as observed in AMD patients. So today, our purpose will be to analyze the impact of lactate transport in age-related macular degeneration. So now I'm going back to the GWAS and the AMD risk alleles in SLC16A gene. The first risk allele that has been identified by the consortium is located within intron 4 here. This common variant is named here intronic risk allele T. Then the consortium looked for rare variants, and so they have found a splice rare variant here, which is predicted to interrupt splicing in intron 2. And so, because the donor site here, the donor site GT, is a sequence present in 99% of human 5' splice sites, we can say that this splice variant is a putative splice site mutation. Nevertheless, this rare allele is not significantly genetically associated with AMD since it did not reach the genome-wide significance. After that, a novel integrative analysis of the results of the GWAS has identified a coding risk allele T or missense risk allele T because the MCT3 variant protein has tryptophan instead of an arginine at the position 235. And finally, independently using induced protein stem cell RPE, risk allele has been identified in the promoter region, and we think that this promoter risk allele could modify the accessibility of chromatin and so could decrease the expression of SLC16AA in AMD patients. So now I'm going to present our experimental models. Since SLC16AA is only expressed in the RPE, we have generated iPS-induced protein stem cell, the retinal pigmented epithelium cells. So thanks to the Professor José Alasael, Dr. Sadek Mansaid, and Professor Isabelle Odo at Hôpital 15 in Paris, we were lucky to get skin biopsy from patients from AMD cohort. And so from the skin biopsies, we have culture fibroblasts, and then we reprogram them with standard viruses to obtain induced protein stem cells colonies. And finally, we differentiate them into iPS-derived iPS cells. At the end, we obtain 5 iPS-RPS cell lines. I'm sorry, 4. So 1 cell line homozygous for the 3 risk alleles, which is promoter, coding, and intronic risk allele. 1 cell line homozygous for 2 risk alleles, with the coding risk allele and the intronic risk allele. 1 iPS-RPS cell line genotype for the splicing risk allele. So it is homozygous. And finally, control iPS-RPS cell line with non-risk genotype. So here, the first result, beginning by the characterization of our iPS cells. So as you see, iPS control, iPS with splicing, iPS with the 3 risk alleles, or iPS with the 2 risk alleles, express endogenous markers of pluripotency, as you see on this immunohistocytochemistry with the expression of the 4 on analog as example, or the result of qPCR with LIN28A or PU54F1. So after differentiation of the iPS into RP, we had to characterize the RP status of iPS-derived RPE cells. And so we had look for expression of RPE markers like RPE65, BEST1, and MITF. And we can find all these RPE markers in the 4 lines here with the control RPE, RPE with the splicing, RPE with 3 risk allele or 2 risk allele. And as expected, there is no expression of stem cell marker here with the suppression of LIN28. Fortunately, we have also the expression of lactate transporters like SLC16AA, and also another one like SLC16A1, which encodes for MCT1. So what is more interesting is that we have studied the expression of SLC16AA mRNA, and we have confirmed that the splice variant is leading to intron retention here. So as you can see, we have intron retention in iPS-RPS cell line homozygous for the splicing risk allele. And of course, we have the splicing. The splicing is normal in control RPE, RPE with the 3 risk alleles or 2 risk alleles. But due to the presence of infant stop codon in the intron 2 of the gene, we have the MCT3 protein is truncated into in its N-terminal domain, and so the protein is not detectable in Western blotting. That's why here we have the absence of NCT3 in iPS-RPE splicing cell here. But fortunately, there is a normal expression of NCT3 in iPS-RPE with promoter coding or intronic risk allele, or with the 2 risk allele. So here, to further characterize the feature of our iPS-RPE, we have performed an immunocytochemistry. And here for the IPS RPE control, IPS RPE with a splicing, IPS with RPE with a promoter coding intronic risk allele, or just with the coding in an intronic risk allele, we have a normal localization of characteristic RPE markers, such as IMTF, ZO1, or ESRIN. And also, as expected, we are looking for MCT1, MCT3 expression. And as expected, there is an absence of MCT3 in IPS RPE with splicing variant, but there is also a normal localization in IPS RPE with the 3 or 2 risk allele. And so we can say that the splicing variant is a null allele. So, what is the impact of arginine 235 tryptophan variant on lactate transport? As I said before, the coding risk allele is a missense variant because the MCT3 variant protein has a tryptophan instead of an arginine at the position 235. But what is interesting is that the residue is located within part of the intracellular loop 6 of the protein, which is specific to primate species, as you see here. So, thanks to the Professor Hyde, who performed a linkage analysis between the SLC16AA risk alleles genetically associated with AMD, they demonstrated that there was an haplotype between the promoter risk allele, the coding risk allele, and the intronic risk allele. But we have hypothesized that the coding risk allele is probably the causative allele. So finally, we have also characterized functionally MCT3 by measuring the transport of the transepithelial lactate transport across IPS-derived RPE cells. And what is important to see is that all IPS RPE cells carrying an AMD risk allele has a significant deficit here of lactate transport, even though expression of MCT3 remain except for the null allele. So today, for the future, we think that the correction of the tryptophan 235 allele back to arginine using CRISPR-Cas9 will be essential to prove that arginine 235 is required for the lactate transport activity. And thank you for your attention.